Okay, it's time to start. Welcome everyone at our weekly colloquium. Our speaker today is Dr. Jonasz Słomka from ETH Zurich. Jonasz did his PhD at MIT. He studied applied mathematics before in Cambridge, and now, as I mentioned, he is in ETH as a postdoc. And today he will tell us about encounter rates involving elongated marine microorganisms. Please.
Okay. Thank you very much. Before I start, I would like to thank the organizers, Professor Fizyki, for the opportunity to give this talk. Since 2018, I'm at ETH. And before I would like to talk about my postdoc, I would just like to spend 2 slides talking about my PhD work. So for my PhD, which was at MIT Department of Mathematics, I work with Professor Jon Dunkel. And we have essentially studied a variant of the Navier-Stokes equation, which was expanded with 2 higher order terms. And this choice is motivated by this type of experimental observations, which represent the vorticity patterns that are created by suspensions of microorganisms. At high enough densities, in a fluid, bacteria that swim, they start to swim collectively, and they generate this sort of vertical patterns. So inspired by this simple statement, we decided to study this phenomenological model, which essentially is in the Landau type Lifshitz model of for pattern formation. Incompressible fluid, correct.
So how is the pressure related if you are missing this?
It's fixed. The pressure is fixed and can be always solved as a...
Pressure in the viscosity because it's the velocity. I mean, the continuity equation, I mean, the very difficulty of an Navier-Stokes equation is that the pressure is a non-local term involving a velocity.
I mean, it is pressure here...
So it's not, it has nothing to do with the Navier-Stokes equation altogether.
This pressure here? No.
No, I mean the whole equation. If the pressure is given, if it's not a function of a velocity, then, I mean, I ask whether this is incompressible fluid.
It is incompressible.
Then if it is incompressible fluid, then I can solve the continuity equation, and the pressure in the Navier-Stokes equation is a non-local
That's correct.
velocity. So, well, then if you say that the pressure is given, then it's...
No, it's not. It's maybe I said it wrong. It's not given by taking the divergence of this equation. I can at each time step solve the Poisson equation for pressure.
But that's not the thing, because the solution of the Poisson equation is local, not this is integral equation. The pressure in the Navier-Stokes equation for incompressible liquid is actually non-local integral of a velocity. That's
As a solution to the Poisson equation, right? Okay, let me continue, because I would just like to spend 2 slides on this. The action of these higher order terms is to create a bandwidth in the Fourier space of linearly unstable modes. There will be a range of modes in the Fourier space that would like to grow exponentially. They get mixed by the advective nonlinearity and are dissipated by the dissipative modes at large and small scales. I don't want to go into too many details. Perhaps the major prediction of my PhD work is that if this energy injected into the fluid is confined to sufficiently narrow range of wave numbers, then the flows that this system creates are of helical nature. This system spontaneously breaks chiral symmetry and produces flows that are right-handed or left-handed, in a sense that the helicity of the total fluid becomes positive or negative depending on random initial conditions. We have also shown that if this energy injection bandwidth in the Fourier space is sufficiently narrow, in this 3-dimensional system, part of the energy injected into the fluid can go to larger scales. If you know a little bit about classical turbulence, that you will know that the energy injected at the large scale, in 3 dimensions, it's supposed to dissipate to smaller and smaller scales. And we shown that we can partially reverse this statement in 3 dimensions with the energy injected at the energy injection scale. Some of the energy goes to larger scales. And this is, if any of you is interested in this, I'm very happy to talk later. But now I would like to talk about my postdoc work that I've done over the past year and a half at ETH. Okay, so after my PhD, I moved to the group of Professor Roman Stocker at ETH, and it's a big lab. I'm a theoretician there. All my colleagues do experiments, and they do experiments on microorganisms that live in oceans. Some of the major questions is that there are countless organisms that live in the oceans, and my colleagues are trying to use microfluidic devices in the laboratory to create conditions that mimic these environmental systems, and they try to understand how these microorganisms respond to various cues. For example, maybe they will be releasing a little bit of food at one part of the channel and see how bacteria find their way up to the source. This would be the typical question. Plankton and bacteria.
There is a tremendous difference of the sizes
Yes.
between plankton and bacteria.
Yes. Bacteria is like 1, 2, 3 microns, and plankton can be up to 100 microns or 200 microns. I'll talk about it in 1 second. So, in order to discuss the results of today, I would like to introduce the concept of marine snow. On the left is shown a satellite picture of one of the Hawaii Islands on the Pacific Ocean. And on the right is the Baltic Sea, a picture taken in the summer of 2010. Hawaii Islands. Hawaii. The left and right, no, that's right. These are different pictures, yes. And I would like you to focus on this is a micro. It does. On the left, we have this red blob here, and on the right, we have this cyan, blue, or green patch. These span hundreds of kilometers, and these so-called algal blooms are visible of space, and what is shown here is the chlorophyll. On the left, chlorophyll. How much chlorophyll there is? There is so much of it that you can see it from space. Okay? And these algal blooms are they arise due to rapid growth, rapid proliferation of microorganisms, of plankton. So this is typical plankton. This would be on the orders of tens to hundreds of microns in size. If the conditions are right, these little organisms can grow very rapidly and form these blooms. If you remember from a few summers ago, we were not allowed to swim in the Baltic Sea because of cyanobacteria. Every year now, I see. Okay.
Actually, the color is pretty proper because in Polish they are called shinica.
Shinica, yes. These are specifically cyanobacteria. Shinica is cyanobacteria. But there is a larger group of organisms that can do that. And they are important for many reasons, and I would like to highlight one concept called biological pump. It has many components, but let me focus on two. Now imagine that at some point these blooms collapse. These phytoplankton die, and because they are heavier than water, they start to sink. So they start to sink through water column, and as they sink, they undergo physical coagulation. They create particles of organic matter, which can potentially sink all the way to the bottom of the ocean. So this is the marine snow. You have a bloom, you have countless amount of phytoplankton that dies and starts to sink, and it's like snow, but just in the sea. So today I would like to talk about this physical coagulation process, try to understand how it works, and also a derivative of this process, which is the interception of these aggregates by swimming bacteria. Because the big idea here is that because phytoplanktons are like plants, but in the sea, they do a lot of photosynthesis, and they can potentially gather together. If they sink to the bottom of the ocean, you can remove the carbon from the atmosphere and store it at the bottom of the ocean. But the amount of how effective this process is depends also on bacteria that can intercept this sinking particle, colonize it, and degrade it, because it's food for bacteria. Okay? So there will be two processes that I'll be talking about today. One is the physical coagulation of phytoplankton as it sinks, and the second is the interception of bacteria of these sinking particles. Okay, so now a little bit more about phytoplankton and its physical properties. Right? So if you look at typical phytoplankton that form these blooms, you will see a variety of shapes. And I would like to understand the impact of the shape of the phytoplankton on this marine snow formation. right? It has been done traditionally as a physical coagulation process, but based on spherical particles. But if you actually look at what these guys look like in the ocean, so this is a sample of water taken near California on the Pacific Ocean, you will see the distribution of sizes, and most of these guys are actually highly elongated. Right? And some typical aspect ratio, length to width. Okay? And some of the key players, so if you have a bloom, the composition in terms of species changes, and you very often see key players called Skeletonema costatum or Trichodismium, and you see that they really long filaments. They can even reach aspect ratio 100. So these are long needles. These are not spherical particles. Right? So I would like to understand what's the impact of shape on this coagulation process. Okay? And to this end, I think it's worth talking a little bit about coagulation theory. So to the best of my knowledge, this was introduced by Marian Smoluchowski more than 100 years ago, where he tried to understand the statistics behind aggregate formation by diffusing colloids. You should have a picture in mind. You have a single small colloid, which is a small spherical particle. It undergoes diffusion. It hits some other particle and forms bigger spherical particles containing, say, I individual particles. And at some point, you have these composite particles containing I individual particles colliding with one that contains J individual particles, and you form a bigger sphere containing I plus 1, I plus J particles. And this process is driven by diffusion, and he also importantly wrote down this cascade of differential equation, one equation for the concentration of each aggregate size. So this gives you the evolution as a function of time of the concentration of individual particles. This gives you the evolution as a function of time of particles that contain, say, 3 individual particles, right?
What are the forces that hold them together?
Once they meet, once they encounter each other. I mean, that depends on the application. For colloids, this would be like van der Waals forces. Not strong.
The proximity force. You don't need any van der Waals. I mean, this is like in the lubrication. If there is a very tiny little channel of a fluid between two spheres, and if the spheres by accident rotate, then they will attract each other and they stick together. That's all in the fluid. We are talking about the particles immersed in something, right?
Okay. So Smoluchowski did it for diffusion-driven aggregation, but the scheme is very general, and it has been since applied to many different aggregating processes, including rain formation. If you have droplets in the clouds that are mixed by turbulence and gravity, they can collide and form bigger droplets. People also use it to model marine snow formation, as I just described, even large-scale structure in a universe, or coagulation in polymers and the gelation transition. Okay, so the scheme is very general, and depending on the process, you have, I should mention, these collision rates depend on the physical process that is driven the aggregation formation. And there are known formulas that quantify the frequency of these collisions. Everybody talks about the so-called collision kernel. For diffusion, it has formula like this. For differential settling, by which I mean spheres falling under gravity.
You don't have the reverse process in equations.
I will not be discussing the reverse process, yes.
The big droplets can...
I will very briefly mention it, but I will not discuss the disintegration process. And also, if you have turbulence mixing, people have historically found the formula for the frequency of the encounters. Yep.
Every encounter is successful?
In this simple picture, yes. But if it's not successful, then nobody really knows how to solve the problem. So what you do, you write a little alpha here that is supposed to represent the collision efficiency, but it's just a trick to express the fact that you don't really know what's going on. So with this in mind, I'm back to marine snow now. And The classical picture of marine snow formation, now you imagine this phytoplankton at the top of the sea and starts to sink. People have traditionally argued that it's the differential settling, so the fact that they are heavy and fall under gravity, as well as turbulent mixing that drive the marine snow formation. It's because ocean is turbulent, and because these guys are heavy and fall under gravity, that's what's responsible for the formation of these clusters. But again, these are rods, these are not spheres, so can I identify formulae like this for shapes that are not spherical? That was my first motivation, and I started with the easiest case possible, which is, let's first think about a single elongated object that is sinking in a quiescent fluid, so no turbulence, but it's elongated. So it's well known that its sinking velocity is a function of its orientation. If you're a rod in a fluid at low Reynolds number, so inertia doesn't matter, a rod that is sinking like this sinks slowest, rod that is sinking vertically sinks fastest, and if you're in between, then your speed is between the two extrema, and also at an oblique angle with respect to gravity. And this is precisely characterized by this expression, which has been known for a long time. These coefficients are functions of the aspect ratio. So now, a simple question. Let's start with a homogeneous mixture of randomly oriented rods and simply count how often they will encounter each other geometrically. And this is in sharp contrast to the spherical case, because if you have spherical particles which are identical and they collide only under gravity, they will never collide under gravity, because they all sink at the same speed. Neglecting hydrodynamic interactions, just the simplest geometrical picture. This is a famous problem in cloud physics, where...
Why should the spheres be different radius?
No, I'm talking about identical spheres now.
Identical.
That's right. Yes, it's known in cloud physics that it would take much longer for the rain to form if there wasn't turbulence, because after condensation you have micron-sized droplets that all slink slowly at the same rate, and it's thanks to the turbulence that rain forms. But now here we have a mechanism. Just because your sinking velocity is a function of your orientation, what's the potential encounter rate? And the sort of first piece of good news is that I was able to find the following the formula for the corresponding encounter rate kernel. And so this is the formula. Again, beta 1 is a function of the aspect ratio, and there are some other key parameters like length of the rod, again, aspect ratio, viscosity of the fluid, gravity, volume of my elongated particle, and the density difference between the particle and the fluid. And I don't want to discuss in detail the derivation of this encounter rate kernel. I'm happy to explain it later. One trick to realize is that if you have identical rods sinking under this expression, then it happens that rods oriented along two different directions, as well as their relative velocity, are all coplanar. But I can maybe explain the details if anyone is interested at some other time. I would first like to describe the consequences of this new formula. So as a function of the aspect ratio, it looks like this. If you are aspect ratio 1, that means that you are a sphere and you never collide. So this is the problem I just mentioned. Identical spheres never collide with each other. And there is a maximum of that maximizes the encounter rate around aspect ratio 50. Okay, and another thing that I can do, I can compare this kernel to the two other previously established collision mechanisms. The first one is differential settling under gravity. The other is a turbulent mixing. And without going into too many details, the point is that here I'm plotting the ratio of these two different mechanisms, and over, I would say, a large range of parameter space, the new mechanism is more efficient.
If you have those roads, and if they collide,
That's right.
not really, I mean, just by the slightly touching the edges, they do not necessarily...
They do not necessarily, that's correct.
So your equation should also have a term which...
Yes. So this is the purely geometrical picture of rods just intersecting each other, and I'm counting a collision when they do intersect each other. But there is
But they
in that
hit them, because if they hit in the corner, they will not coalesce.
that depends on how sticky they are, right? That's a separate problem, and I agree with that criticism. But if you look at the literature, it's typical to first just count the geometric collisions, and then think about what happens if you actually encounter each other, how efficient this contact process is. And the second part of the question I'm not addressing when I'm writing down this formula. And it's the standard way to do also for the case of spherical particles. You first think about some process that is bringing particles together, and then you separate it from the efficiency of the actual contact. What's the probability that this actually is going to result in collision? So since then, I made a little bit more of a progress, and I've also tried to understand the collision rate between two different types of particles. So I have red and blue particles. One has length, aspect ratio, and its own density difference. Then the other species also has its length, aspect ratio, and the density difference. And you can also ask the question, what's the collision rate between the red and blue species? And it turns out to be also possible to find the collision kernel analytically, at least in the limit of really thin and perfect rods. So here are the way I tackle this problem, and again, I would just state the result, is that you can expand the collision kernel as a power of the inverse aspect ratio. So for very thin particles, this is the dominant term. For the perfect rods, you can still find the analytical formula. And for rods of finite width, you have to do numerical simulations, which are very simple. The point is, if you have two types of rods that differ by length, aspect ratio, or the density difference, I claim I have a formula that will tell you the geometrical encounters in a quiescent fluid. So now I can go back to the Smoluchowski equation and think about what are the consequences of this new formulae. Okay, so again, on the right-hand side, I have a collision process that is driven by differential setting under gravity and under turbulence, and they combine effect. And on the left, I would like to think of a collision process and use the new kernel to study it. So I will make one more assumption, which is the following. Now, my particles that collide with each other are perfect filaments. And I'm assuming that when they touch each other, there is some mechanisms that makes them into a bundle. A bundle that has the same length as the individual filament, but simply whose width grows. So you have sort of two spaghetti filaments that touch each other, and there is some mechanism that brings them together and aligns them into a nice bundle. Exactly. So here is my aggregate size, and I stop at when my bundle reaches aspect ratio like 1.1. It's still fat, but this is already the spherical limit. Okay, that's a good point. So here is the combined kernel as a function of. So like the point here gives you the collision efficiency between particle containing 100 filaments and 1,000 filaments. And one key point is that I would like you to appreciate that this kernel is concentrated in this corner of the sort of phase space, meaning that the prediction from this new formula is that under this collision rule, it's actually the thinnest and thickest bundle in the system that collide most often. Okay? Unlike, for example, the impact of turbulence or the combined impact of turbulence in differential setting for spheres, because the kernel is more concentrated around similarly sized particles. One more small comment, because if you think it's a little bit controversial that I take two filaments, let them align together to form a bundle, this is an example of trichodismium. And in the ocean, it's very often observed to form bundles like this. And I have a colleague who studies them in a lab. This is now on the bottom of a petri dish. You can see that these filaments, under certain environmental conditions, can actually form these bundles. Okay, so this is not just a theoretical possibility. Some of these organisms actually realize this collision rule.
Here's the living organism?
The living organisms, yes, yes. So, back to the Smoluchowski coagulation equation. Now I'll be simulating this set of ordinary differential equations. So the physical picture you should have in mind is the water column of thickness about 100 microns that contains different concentrations of these filaments undergoing these collision rules. The individual filaments in my system grow, they divide. So this provides the source of the particles in this fluid layer. And all of my particles sink at some rate, so they are removed from this 100 meters layer. And in between, they undergo this coagulation through this nonlinear process. So the classical picture is on the right-hand side. The concentrations of spherical aggregates of different size eventually always settle on time-independent concentrations. Okay? Meaning that here my individual hypothetical spherical phytoplankton keeps growing because it divides, and it creates filaments, aggregates of larger size. They all sink, and this process is perfectly balanced in the sense that the concentrations are time independent. Now, for the equivalent system of rods, I've observed the following scenario. The system reaches statistically stationary state, but it's not steady. It's not time independent, meaning that the concentration of individual filaments and the composite bundles oscillate as a function of time. So I think this is one of the most useful predictions because it says that now, if you have those phytoplankton that is elongated in the ocean, that the prediction is that if its growth is balanced by sinking and coagulation, the concentrations of the bundles will oscillate by even an order of magnitude on a scale of 1 week. So the prediction is that if you go to the ocean and see those bundles, you should see them oscillating as a function of time on a scale of a week. And a quick comment to this end as well. You asked me about the disintegration process. Just a general comment about Smoluchowski equation, the coagulation equation. It's 100 years old, but as far as I know, it has only recently been discovered that these type of systems can sustain oscillations in a steady state. And I try to argue that the system of sedimenting rods is actually a concrete possible realization of these oscillations in coagulation dynamics. Okay. Do you have any questions about this first part, the coagulation process between rods at this stage? Okay. So this is the sort of the end of the first part of the talk, and now I want to go back to the biological pump and talk about bacteria. So I've just tried to describe this physical coagulation between elongated phytoplankton, but now I want to be interested in this later process when my bundles have already coagulated. I have some large particle that is sinking, but now I have a sea of bacteria around it that can swim, and they can encounter this particle.
I'm slow, so I'm thinking about your roads. If the roads move in the fluid, they paddle. So they disturb the fluid considerably. And because of there's a very different thing when the sphere rotates in the liquid. If you have two spheres, they are rotating, then the interaction is basically by the macroscopes, right? And if it's paddle, it's completely different.
Yes.
So
That's not included.
the cross-section for the collision depends on this change of the flow. And
Yes.
It's also isotropic.
Yes, so
If I have two roads, they try to collide in the liquid, then I would assume that the cross-section for this process depends dramatically on the angles.
that's right. So, I mean, again, I am neglecting hydrodynamic interactions between these particles. This is neglected. This is this.
Lots of them. I mean, if you have a few of them, that will be, of course, very
Yes.
reasonable to assume. But if you have lots of them, so they form these clogs and packages or whatever, then I would say that this hydrodynamic interaction is important. We have a group in Warsaw which does a sedimentation of the particles at the Institute of Fundamental Research, and Professor Rekielewska is an expert on it. And they claim that the hydrodynamic interaction is predominantly important for the sedimentation, and the sedimentation is very much similar to what are you doing.
Yes, yes, that's correct.
This is sedimentate, right? In some places they are hard tires, and in some places with some bacteria. So but that's the sedimentation problem. And I'm worrying why in any case, since we are in water, we should talk to them.
Yes.
Particles are treated as
W piątek, tak. Okay.
treated as test particles in electrodynamics now. So the back reaction is another problem.
Yeah.
To some extent, he's neglecting a lot, but when
just-
people are doing experiments, they can observe what he's showing us with the equations.
Precisely what we do in electrodynamics. When we consider the motion of test particles of a dust.
Yeah. Let me continue. Thank you. So now let's. Now I would try to, in the second process, I will be a little bit more careful, and I would try to talk about the impact of hydrodynamic interactions, about fluid shear, actually. So now, by some process, my phytoplankton got together, and it's a big clump of material that is sinking. And there are swimming bacteria that can intercept it. And they can eat it if they find it, so it's important to quantify how often this happens. So I would like to briefly just outline how this has been traditionally modeled. Okay? And the traditional approach is very simple, and it's good in its simplicity, but it boils down to the following problem. If you look at mottled bacteria under the microscope, at sufficiently long times, at sufficiently large scales, they essentially perform like a Brownian motion, because they usually swim forward, and they have some internal mechanism to randomize the redirection of motion. So they swim forward, then they tumble and swim in some random direction. If you wait for long enough, this will be just a Brownian motion with some effective diffusion coefficient. So if you accept this, then people have traditionally said, well, if I have bacteria swimming around a sinking particle, it's really just like a mass or heat transfer to a spherical sphere by some diffusing colloid. In which case, the problem boils down to solving advection diffusion equation. You have advection diffusion equation with a diffusivity that is the effective diffusivity of bacteria. And then you solve for the diffusive flux onto your particle, and again, D is the effective diffusivity, which is larger because these bacteria can actually swim. Okay? So this has been the traditional approach, but if you go to the ocean and observed how many particles there are of a given size, so if you look at the size spectra of the particles, you will see the picture varies from the depth or the geographical location, but the point is you always see some sort of power law, and you will quickly realize that the number of particles that is largest are the smallest ones. Most of the particles of these sinking particles are actually below 100 microns. In which case, the encounter process between bacteria and these sinking particles is not ballistic anymore. It's not diffusive anymore. Because, as I said, bacteria are swimming straight, then randomize the orientation of motion. So there is sort of like dx, the grid size on which they walk, is typically tens to hundreds of microns, is as big as the sinking particle. And for this reason, the encounter process is not really diffusive, it's ballistic, meaning that on the scales relevant for the encounter process, bacterial trajectories are more like straight lines. That's what you should think about. However, additionally, we know that because the particle sinks, it creates a disturbance in the flow. This can also change bacterial orientation by shear. Okay? So now the question for me was, okay, what's the impact of elongation and shear for the encounter rates between bacteria and sinking particles in this ballistic range? And the model to do that is now It's very simple and very classical. Now, let's forget about the details of the shape of the sinking particle. It's some large blob. Okay, so let's take it to be a sphere, my spherical cow. Okay, and the flow field that it creates is given by the Stokes flow. This has been known since 19th century. And I would take my bacterium to be now elongated rod that can swim straight at velocity UB, and also it responds to the flow around it, as well as its direction depends on the fluid shear through the so-called Jeffrey equation. So if you like, I have a large sinking sphere and a small bacterium that is like a little submarine that can swim straight, can be also advected by the flow, but whose direction of swimming can also be changed by the flow due to particle. Is this model clear? If you've never seen Jeffrey equation, this is an equation for a small ellipsoid that responds to the shear of a fluid flow. These are the symmetric and antisymmetric components of the velocity gradient tensor. You have a velocity, you compute the 3x3 matrix of velocity gradients. This is its symmetric part, and this is its antisymmetric part, and this is a parameter that depends on the aspect ratio. So before I do the hardcore simulation and start with a random swimming bacteria with random orientations below my sinking particle, I wanted to first get some intuition about what is the action of fluid shear on the orientation of small ellipsoids around that big particles. Okay, so it turns out, so this is my sinking particle. Okay, the flow goes like that from bottom to top, and I'll be putting a small spherical or elongated particle at each point in that fluid, and see how it wants to orient. Okay, so long story short, the response of rods breaks the sort of upfront and backward fore-aft symmetry of the flow, meaning that small rods around a sinking particle first want to, if you put them like this here and hold them by the center of mass, they will eventually reorient in the plane out of the picture. If you hold it here, they will always spin like this in the plane of the picture. And when you put them to the back of the particle, they will always point along these white lines. So the shear really orients little rods differently upstream and downstream of the particle. Okay, and to actually show this, I convinced my colleague to do a simple experiment. So he put a spherical particle in a microfluidic channel, so a small channel, and flashed a suspension of plankton, which is elongated. So this is still non-motile, but it's an elongated particle that is just simply advected around that big sphere. And he measured at various positions their orientations. So each dot represents the position and the orientation measured, and you will see that. So yellow represents position like this, and blue represents vertical position. And you will see that, and all of them are down at the bottom in the experiment, they come in with random orientations. So indeed, the fluid shear orients them tangentially or horizontally upfront of the particle and radially towards the back of the particle. And now when I simulated the full system of passive rods, so still not swimming, but responding to shear, this is the picture that looks very similar. So
we are looking at the two-dimensional picture, but you already have
that's right. That's right.
So this yellow thing is actually a stagnation point in a sense. I have a sphere which moves, so there is a stagnation point.
Yes.
And then, of course, the gradients of velocity changes in the front and in the back.
That's exactly my point. That's right. That's exactly my point. Yeah. So yes, both in the simulations, and I have typically maybe rods arriving like this, and I'm looking at the XY projection of that rod. Okay. So now, this is how I understand the behavior of small elongated particles around a big sphere. Okay. So now to the full encounter problem. Now we can do the full simulation of swimming bacteria that respond to the flow around the sinking particle and also respond to the shear around the sinking particle. And I can measure the encounter efficiency, which is like the probability of the attachment, how effective is the collection process, as a function of the flow speed. That's right, as a function of the flow speed, how quickly the particle is sinking. If you turn the shear off, you have the green curve. If bacteria were spherical particles, you would have the blue curve. But now elongated particles, they respond much more differently. In particular, for low sinking speeds, they are the most efficient shape to maximize the collision efficiency. But this efficiency drops very fast, and they are suddenly really bad until your flow speed is so high that swimming doesn't matter and you're just a passive particle. Okay? So why this rapid change in the response? And now I want to rationalize it in terms of the picture of how the shear reorients you as a swimming particle. So this is a simulation, an example of a simulation of the full process now. You may think that the easiest way to catch your sinking particle, right, is if you're below it and it's falling onto your head and you just want to swim up, right? So you'll be swimming up. Okay, this is my particle. You'll be swimming up. But remember what the shear does. Shear turns you tangential to the sinking particle. You keep on swimming, and therefore you keep missing the target. This is actually a very unlikely position, which is counterintuitive, to intercept the sinking particle. So this is one mechanism, okay? And now another mechanism that actually results in very frequent collisions is your particle is sinking, you start far away from the sinking particle, oriented horizontally, the particle passes by you, you skip this screening region below, but now to the back, you remember, shear wants to orient you in the right direction already, and you hit the particle from behind. So now,
Because the second particle becomes faster and can
yes, because there is
somehow pulled.
yes, it's exactly. It's somehow sucked into the back. Yes, exactly. So now back to this encounter efficiency curve. The reason why elongated swimmers are most efficient for slow sinking speeds is because they can realize the sucking to the back. But then as your sinking particles start sinking faster and faster, you can only hope to attach directly from below. But then you have only the screening mechanism available. And that's why I have this gap where you have essentially no encounters. And that's essentially the impact of shear on the encounter process that I wanted to talk about. So I'm almost done. I still like to very briefly point out some other projects I'm working on. So as I told you, I have a friend who is studying tracheodismium, these filaments that can, when they come together, they can form bundles. And I try to build simple models that can realize this state. I also have a cool experimental colleague with whom I started to do experiments and simulations of the motion of fibers in a flow, just to understand how it rotates, inspired by all these strange responses of fibers around a sphere. And also with Maciek, we've recently also started to think about how to generalize this encounter picture to the case when the rotating sphere, the sphere is falling, but it's also spinning, to see the impact of spin. Okay, so to conclude, what I've worked on in the past year and a half was to try to understand the geometric collisions between identical and dissimilar rods, as well as the encounter process between bacteria and sinking particles, the impact of flow and shape. Okay, thank you.
So thank you very much for this talk. We have some questions.
Very nice talk, thank you.
Thank you.
I'm sure that the answer will be yes, but maybe you will surprise me. So since you already checked the impact of the elongation, and it has a profound effect with some respect, especially that you show that the highest probability of encounter was actually for the same size particles, not for different size particles than differently in the spherical case. So why won't you also study the impact of bundles flowing like that with the bacteria trying to feed on this? Because in the second part you studied the spherical particles still,
Yes.
right? So can you do it also for kind of a flowing bundles of filaments and see the difference?
Replace this with a
Yeah.
bundle. Yes, but it feels very difficult. Like, I don't know simple expressions for a flow around a cylinder.
Right, so this is basically because it's much more complicated
Yes,
numerically.
yes. The fluid flow is very complicated. It would have to be, I think, directly go to numerical simulations
Right, so
for the
answer is
first to understand the flow.
Okay, thanks.
Okay, do we have more questions or comments?
You didn't say anything about this disintegration. I mean, if I have this bundle, I mean, actually in the Smoluchowski equation, there is a term for disintegration, right?
You can include it. He didn't do
Yeah.
that originally, but one can do that, yes.
Yeah, but this, I'm not talking about the original paper,
Yes,
but
yes.
what is called Smoluchowski
That's right.
equation, or coagulation.
No, it's definitely a good way to generalize and make it.
And they don't break these bundles?
So these guys can do essentially everything. They can you can think about it that they can control both the encounter and the disintegration. This is so my colleague
Because
does something to them so that they want to stick together. But if you change some conditions, they want to disintegrate.
The reason why I'm asking is that one of the use of the Smoluchowski equation with this both coagulation and disintegration terms is that you can form a critical size object in the thing, right? I mean, the people try to use it to describe the condensation of a vapor or whatever, right? If you have a coagulation, then the particles can get to the critical size, become stable, and when it's slightly smaller, they disintegrate. When it's slightly larger, then they start to grow. And in the linear version, that's exponential growth. So that doesn't happen with these blobs of the marine snow? That it
I don't know.
forms the islands or whatever?
It's a good point. I don't know.
That's very nice work.
Thank you.
Okay, as there are no other questions, let's thank Jonasz once again.